in talking about modification of RNA, right, RNA's processing or modification, that is one way, in kind of roundabout way, to actually regulate translation. Okay, so translation has its own regulatory mechanism, but again, in terms of processing is partly involved with the regulation of translation. So they're kind of meshed in. Okay, so that's why this next the 3 sections for this set of exam combine RNA and RNA processing with translation. So, no question? Okay, so let's see. Sure.
So for chromatin remodeling to occur, you have to have the histoid or the acetylation of the histone tail. And that depends on what needs to happen in the cell, which lysine residue is modified.
If we're only talking about acetylation, remember we only focus on acetylation, but a bigger picture modification of the histone involves activities other than acetylation. But as far as this class is concerned, yes, acetylation. And as far as we're concerned, it's acetylation of the histone tail. The histone core, so the histone actually, yes, it's more complex, but as far as this class is concerned, that's all that you need to know is acetylation, the pattern of acetylation of the histone tail.
Okay, and so how does that cause it to like really unravel? It's like, how does it make it really
Whether the histone tail bind or not bind, if it binds tightly or it doesn't bind tightly. So the whole idea is really has to do with how tightly the histone tails hang on to the DNA. If it's hang on tightly, then the DNA won't become naked and therefore no expression. If the histone tails does not bind tightly, then there's a chance of unraveling to occur, and then you have the naked DNA, right? So it's all about binding. That's it? So that you don't seem convinced, but
is that dependent upon whether or not you get the trithorax and the polycomb from your parents?
that's in addition.
Okay.
That's in addition.
Okay.
Yes. So I depended on you to make that connection, and you just did. Right. So if you have the polycomb, so the idea is this: is if you have the euchromatin structure, this is it, right? And here's the DNA that is exposed to the transcription factors that can regulate the processing of the histone code by recruiting enzymes or not recruiting enzyme, and to carry out basically the histone acetyltransferase to carry out acetylation of specific lysine. So far so good? It's exposed. If the polycomb protein is somehow involved, then that causes further folding, so that DNA sequence is no longer there. So regardless of whether you have transcription factors, tissue-specific transcription factors or not, you're not going to be able to cause recruitment of enzymes, and therefore you're not going to be able to change the histone code for chromatin remodeling to occur. There you go. Right? And if the trithorax protein is there, then it prevents any further folding, and so that piece of DNA is still exposed. Now, still depends on tissue-specific transcription factors to bind to the specific DNA, even with the trithorax protein, right? The trithorax protein, all it does is it keeps that regulatory DNA, the response element, exposed. But you still need the tissue-specific transcription factors to bind to that to cause chromatin remodeling. Easy. Right, it's active, now it has to turn on. Right, so the difference between being active and turning on. Right? Yes. Good? Right? That makes sense, right?
Yes.
Yeah. Okay.
Sure.
if you don't know what I'm talking about, you are in trouble, honestly, because that's the type of things I need you to be able to understand, right, from this set of exam, okay? That type of regulation, it's just, I talk about different regulation, but they all, they're all integrated, right? They're all integrated. Okay, so the next set is the next. Can you see in the back? It seems like it's a little bright. Okay, all right. Cell-specific regulation of mRNA production. Okay. Are we? Is this the right place? Yeah. Okay. Yeah. So then we're going to talk about basically. Sorry, give me a second here. I'm looking at some of the slides and looks like I might be okay. Let me flip through. Oh, okay, just I know what's going on. Okay, so I have the order of events different. So we're going to continue. Actually, we're not going to talk about modification of RNA yet. We're going to continue with talking about translation. Okay, so transcription, translation, a little bit of review, and then we'll come back and talk about modification later. Forgot that that's how we built the lectures. Okay, so remind you that you have a gene, and then to carry out, use as a template to carry out transcription, and with all the modification that we will talk about later, then the mature mRNA will be used as a template to carry out amino acid polypeptide or protein synthesis or polypeptide synthesis. And then you have translational modification to actually get a protein. Okay. Just remind you about the genetic code. Don't have to memorize this. I'm not going to require you to be able to use this or anything. I assume that you remember and you will make 100% on this if I ask you today. So I would leave it for you about wobble bases and all of that. Won't ask you about any of that. Okay, this class is about regulation. Remind you about the synthesis of a polypeptide. Okay, translation is the synthesis of a polypeptide, not a protein. Again, translation is a synthesis of a polypeptide. A polypeptide needs to undergo processing or modification before it becomes a protein. Okay, and so the synthesis or translation then is carried out by having the ribosome reading the mRNA template from the 5' to 3' direction to synthesize an amino acid sequence from the amino terminus to the carboxy terminus. And obviously amino acids are required, right? So in the cytosol you have a different amino acids present so that amino acids can be attracted to the area. Of course, the amino acids have to be bound to tRNA. That's where tRNAs come in. Okay? So again, this should be a review for all of you. So again, translation. What does translation do? Translation determines the primary structure only. Right? That's what translation does. Translation, at the end of translation, you get a polypeptide, which is an amino acid sequence in the primary structure. That primary structure is going to be folded into a secondary structure, and the two organized secondary structures are the alpha helix. There are pictures later on, and beta-pleated sheets. You also have random coils, right? Random coils is just the extra amino acids that don't either fit into the alpha helix or beta-pleated sheets. They're just the leftover, okay? But again, the two major organized structures for the two organized secondary structures are alpha is an alpha helix and beta-pleated sheets. Tertiary structure is when you take the primary structure into secondary structure and then further fold it into a condensed polypeptide. Usually, that is when you refer to a polypeptide as a protein. Right? So a protein, by definition, is a properly folded and functional protein. In order for a protein to be called protein, the polypeptide has to be properly folded and functional, right? And we're not talking about mutations or anything like that. And then quaternary structure is when you have more than one tertiary structures interacting. Now, later on, we'll talk about subunits. Subunits, multiple subunits interacting is different from a quaternary structures where you have multiple tertiary structures interacting. For instance, an example would be a hemoglobin. Hemoglobin requires two hemoglobin alpha and two hemoglobin beta to interact before the whole thing can function. So each hemoglobin alpha or beta doesn't do anything, right? So the only way that hemoglobin can function is as a quaternary structure, as four of these units together, and they are bound by disulfide bonds, so a covalent bond. So far so good? Which is different from if you have a complex of proteins that must function. For instance, RNA polymerase. RNA polymerase is composed of roughly 12 subunits, but they're not bound by covalent bonds. They're only bound by non-covalent bonds, hydrogen bonding, hydrophobic interaction, van der Waals interaction, and ionic interaction, right? Then an RNA polymerase is not a quaternary structure. So again, by definition, a quaternary structure is when you have these tertiary structures forming covalent bonds to one another. So they can't escape. There's no way that these units can escape from one another, whereas a protein complex like RNA polymerase, these subunits can come together, but then they can also break apart. That's the difference, okay? And then let's look at how these structures are formed. So again, an example of the primary structure, and if you remember, the primary structure is you have a peptide backbone, right? So you have the peptide bonds, and then the R chains or side groups, or R groups or side chains point outward, okay, either in this direction or that direction, or toward you or into the board, okay? In order to form the secondary structure, hydrogen bonds are formed within the polypeptide backbone. There you go. Okay? So you notice, so in order for the secondary structures to form either alpha helix or beta pleated sheets, you notice that the hydrogen here forms a hydrogen bond with that O here, and both of them belong to the polypeptide backbone. The R group, right, aren't involved. R groups are not involved in the formation of secondary structures. Okay? Whether beta pleated sheets or alpha helix, you notice this again. This is the interaction within the polypeptide backbone versus a tertiary structure is when you actually have side chains interacting with one another. Non-polar side chains interact with non-polar side chains. Positive or basic side chains would interact with negatively charged or acidic side chains, right? That's so again, tertiary structures involve the interactions among side chains. Also, this is where you can also find the formation of a covalent bond, the disulfide bridge. The disulfide bond is the only covalent bond found in a tertiary structure. It is also the only covalent bond found within a quaternary structure. Okay. And of course, quaternary structure also involves the other bonds, hydrogen bonds, van der Waals interaction, and so on. Okay? So again, I'm assuming that most, if not all of this is a review for you. We're not going to harp on it. Now, so the example of the hemoglobin is just that. Okay? Okay. I forgot I had that section in there. So we went through roughly the regulation of transcription, and then we talked briefly about translation. Now let's look at modification of the mRNA, and then which leads into regulation of translation. The two go hand in hand. Okay? You see this code post? Forget about it. Don't worry about that. Some modification of code, some of post. Don't worry about it. Just call them transcriptional modification is okay. Right? Transcriptional RNA modification or transcriptional. That actually is redundant. Transcriptional modification is sufficient, or transcriptional processing is sufficient. And we're going to talk about 3 different types. We're going to talk about capping. What is capping? Which is fairly simple. It'll take 2 seconds to talk about. But then, what's the functions? What are the functions of capping? We're going to talk about polyadenylation, and then we'll talk about the functions of polyadenylation, and then we'll talk about RNA splicing, which will, well, all 3 of them would take a while to talk about. Okay. So if you were to have a primary structure here of mRNA, so we in the previous slide we call this either the primary transcript or the nuclear RNA, right? Which include coding and non-coding regions, sequences for translation. Then the 5' end would be capped. Okay. And capping is basically the addition of a guanidine phosphate, guanosine phosphate. That's it. Okay. So the cap is there. The difference between the addition of the unique thing about capping is, number one, that the addition of the nucleotide is to the 5' end, which is the only time when nucleotides are added to the 5' end of a nucleic acid. Number one. Number two, the addition is a 5' to 5' interaction, instead of a 3' to 5' interaction to form a phosphodiester bond. Okay? And again, I assume that you know what I just said. You should have learned that in Bio 2. Okay? So again, those are the 2 unique features about capping. Is number one, again, is the fact that a nucleotide is added to the 5' end of nucleic acid. And number 2 is the fact that it's a 5' to 5' interaction. So that's capping. Okay? And so if you, let me see if there's a better picture. No, okay, that's all. Sorry, that's all that you get. So we took a bunch of pictures out just to make things simple. Imagine that if you were to have a DNA template here and an RNA polymerase read it going this way and then synthesize that primary structure. As soon as the 5' end is synthesized and as transcription is continuing, that capping event occurs. That's the very first event that occurs. So modification occurs during transcription. Okay? And the next thing that occurs is then you would have removal of introns, which is RNA splicing. We'll talk about RNA splicing at the end. Okay? So we'll just move forward and actually prokaryote. I keep pointing up that way. We'll go in here, and then we move all the way to the end where polyadenylation occurs, which is the addition of a series of a couple of hundred of adenosine phosphate are added at the 3' end of the transcript. So far, so good? Okay, and that's what happens. So let's look at let's look at capping. What are the functions? And this is not even there. It's missing a couple of functions here. Okay. In the nucleus, the cap has functions in both the nucleus and the cytosol. In the nucleus, the cap, what does it indicate? The cap indicates that this is an mRNA and not any other RNA. Most, I'd say 99% of the mRNAs that are made in your nucleus are capped. There are some that are not capped, but like I said, 99% more or less are capped. Okay? So then by seeing a cap, the cell knows that this is an mRNA. And where do mRNAs, where should they go? They need to go to the cytosol. Okay? So that's the second function of the cap, is that the cap acts as a marker, as a signal for export. The cap tells the cell that this mRNA that is in the nucleus needs to be delivered to the cytosol. So that's the second function of the cap. So far so good? Now, remember the cap is added first, right? The cap is in, just cross this out, pretend that this is a primary transcript. Cross that out. So the cap is added first. So as the cell continues, the third function of a cap is the cap is involved in choosing and in the removal of the first intron. Okay? And it's a little complicated. It's not as easy as removing the first intron. Sometimes the first intron, as well as some other pieces of the coding region, are removed simultaneously. So again, the cap helps the machinery choose which intron and which piece of RNA that needs to be removed through RNA splicing. Okay? So that's the third function of a cap. All of these are functions within the cell, within the nucleus. The fourth function is the cap, if an mRNA has a cap, is involved in the regulation of the initiation of translation. Again, the cap, once in the cytosol, the cap is involved in the initiation, in the regulation of the initiation of translation. Okay. So, if you have a cap, then initiation of translation occurs. If you don't have a cap, then you don't have the initiation of translation. That boils down to that. Okay. That's the fourth. And there's something, one more thing, but we'll talk about it when we talk later on about regulation. Yes, sir.
Will all mRNA have the cap? Like, will there be any times that the mRNA can be produced without a cap?
Like I said, 99% roughly of mRNA do have a cap, and there are some that don't have a cap. They need something else.
Okay, so what happens to the mRNA that makes it into the cytosol without a cap?
I will tell you. I will tell you. It's actually in here. I already told you, but I will repeat it orally. Yeah. But yes, we'll go through all of that. I don't mean to make light of that, but lots of stuff to talk about. Okay, so that's capping. You know when it's done. You know roughly what capping is, and then most importantly, functions. Okay. Poly-A tail. Again, we'll leave RNA splicing for last. We'll leave RNA splicing for last. Poly-A tail. Once the entire, so here's the entire primary structure right here. Let's say primary transcript right here. Okay. There's a site that is referred to as the polyadenylation site that is found in the middle of, well, maybe not right smack in the middle. This is not to scale, by the way. It's somewhere within, the polyadenylation site is somewhere within the RNA, right? The primary structure, primary transcript. It's not found in the DNA. It's only found within the primary transcript. That polyadenylation site indicates where polyadenylation must occur. In this If that were the polyadenylation site, then polyadenylation occurs here. And roughly, we're not going to go into a lot of details, but polyadenylation occurs when the RNA itself is cleaved off. So it's separated. These two regions are separated, and then the A's, the poly A tail, is added to that 3' end. And you notice what happened to this? It's gone. It's no longer a part of the mature mRNA. Right? And because they're separated, that's termination of transcription. That's it. You can't carry out transcription, can't continue with this strand any longer, right? So by definition, polyadenylation, right, induces termination of transcription. So therefore, that's why there is not a transcriptional termination site. There doesn't have to be one. Okay? Because again, by definition, if the cell undergoes polyadenylation, then transcriptional termination occurs. That's it. Yes, ma'am.
What is cleave?
Enzymes that cleave. What is cleave cut?
Yeah, but what is,
What enzymes that cleaves it?
no, what is, what goes under cleavage in this stage?
What goes in the-
being cleaved?
I think
Yeah,
she wants,
what-
what's the
Oh,
OH?
the
Yeah.
transcript. The primary transcript is cleaved. Right? That's the primary transcript right there, and the primary transcript is the polyadenylation site. Thank you. I'm sorry. The polyadenylation site is found in the primary transcript, and a part of the first part of polyadenylation is the cleavage at that site of the primary transcript, and then the second part is the addition of the poly A tail. Yes, ma'am.
So by the time the 3' end already gets polyadenylated, the 5' end's already been capped?
Yes. Long before. Okay. So far so good. So now you have by the time that the primary transcript or the mRNA now, we can call it the mRNA, is not quite mature yet, but an mRNA is polyadenylated, processing continues and wrapping up, right? What kind of processing? RNA polymerase, RNA splicing continues. Okay, so by the time that polyadenylation occurs, you still have maybe 1 or 2 introns that still need to be removed. Okay, which is let's go with the function of polyadenylation first, and then come back to the previous slide. So which is one function of polyadenylation? Yes, ma'am.
Is there multiple polyadenylation sites?
We'll talk about that. That's why I just said it. I will do this, and then we'll come back to that slide. Okay. So let's talk about the polyadenylation site first. So if you have polyadenylation, and I just told you that once polyadenylation occurs, RNA splicing is not done yet. So one of the very first function, the first function of polyadenylation is to induce transcriptional termination, right? That's the first one. The second function is really the poly A tail. Poly A tail helps aid in determining the splicing of the very last intron, or the last piece of RNA that is removed. So the cap helps RNA polymerase, RNA splicing at the beginning, and poly A tail helps in the RNA splicing at the end. Okay? Nucleus transport. In addition to the cap, and like I said, 99% of the mRNAs contain a cap, 100% of RNA contains a poly A tail. So along with the cap, the poly A tail is also needs to be present in order to signal that this mRNA needs to be transported or delivered to the cytosol. So both the cap and the poly A tail. What happens if you're missing the poly A tail, or you're missing a cap that is supposed to have a cap? Then no delivery, right? No transport. Okay. All 3 of those so far are in the nucleus. Let's go out. Let's go out into the cytosol. In the cytosol, again, together with the cap, the poly A tail is going to help in the regulation of the initiation of translation, what it just says here, right? So this is in the cytosol that the poly A tail does that, and I'll explain how that works. And then lastly, the poly A tail actually also controls the amount of translation. Remember, last Thursday we had a slide that said that shows if you have a stable promoter region is open, then you would get more transcripts. And then if you have increased number of transcripts, you would have a lot of proteins or polypeptides made. And if you don't have a lot of transcripts, you don't have a lot. However, one way to get around having to make a lot of transcript and still have a lot of protein made is by making a much longer poly A tail. Okay, so the longer the poly A tail, the higher the amount of translation. Therefore, the higher the amount of polypeptides that will be made. I will show a slide and we'll talk about that even more. Trust me, we'll do that. You're going to get sick of it. So that's capping and polyadenylation, and those are the functions. We're going to mash the two together at some point, and then now show you how control is done, how RNA processing is involved in the regulation of translation. You already see hints of it. Are we good so far? And as promised, I'm going to go back to the previous slide. The polyadenylation. On an RNA, there may, does not always happen there, but there may be multiple polyadenylation sites. So the example here is that there are 5, right? There's one, let's see, one here, one here, one here, one here, and one here, right? There are 5. And the cell gets to choose which polyadenylation sites it gets to use, okay? But the very simple question is, so what? Why does it matter which polyadenylation site that's used? What does choosing different polyadenylation sites, again, very simply, what immediately is the effect?
Termination of the transcription.
Well, more than, by definition, termination, but what is the effect in addition to termination? The length of the mRNA.
Oh, okay.
Right? If it uses this, then you get a shorter. In fact, you don't have that whole exon, and you're missing a part of that exon. If the cell uses that, then that whole exon is used, but you're missing all these exons, right? Not only that, but if you use that polyadenylation site, then that poly A tail will help in the RNA splicing of the previous intron. If the cell uses that polyadenylation site, then that poly A tail will be used to splice that intron. So the regulation of which intron to remove, right, which portion of the RNA to be removed is also determined by which polyadenylation site that is used. But the immediate, again, the most important effect is the fact that depending on which polyadenylation site you use, that will determine the length of the RNA. And the length of the RNA, therefore, equates to determining the length of the coding region, as well as the length of the 3' UTR. Right? Has nothing to do with the length of the poly A tail itself. The length of a poly A tail is determined by other things that we won't even talk about in this class. Are we good with that? With the fact that there are multiple there's only one capping site. There are multiple polyadenylation sites. What does that tell you about variability? Going back to the first set of examples, variability is important, right? In terms of, remember that we have more regulatory DNA than any other organism, and we're about to encounter how that equates to variability. right? So you can go from one gene and you can make at least 5 different 5 different proteins from that one gene. That's us, versus something like a prokaryotic cell, maybe some lower animals that can't do that. Right? So already you're seeing variability. Okay? So we're good? Yes, ma'am.
So you said whatever poly A site is next to a certain exon or intron, that will determine what is used?
No, it's just whichever polyadenylation site is used, that determines the length of the mRNA. That's just it. And it's shown here. Right? I think it's pretty clear that it's shown here. If you use that, you lose all of that. And this is important. It's regulated by external signals. Okay. So rough idea, just rough, is you would have a cell carrying out expression of this gene by having tissue-specific transcription factors binding to regulate chromatin remodeling, and then expression of this gene, and then remember, transcription factors only regulate transcription. Has nothing to do with modification of RNA. So then other regulatory proteins have to bind to regulatory, not DNA sequences, but regulatory sequences within the RNA itself. Right? So structure function, if a structure is a function is important enough, the function is conserved. If a structure is important enough, it is conserved. If a mechanism is important enough, it is conserved. So the idea of regulating activity by binding to regulatory sequences is conserved in this case. You have regulatory sequences in the DNA, you have regulatory sequences within the RNA. Does that make sense? So we're going back to the idea of conserved structure and conserved function, and then structure-function relationship. Yes, sir?
You mentioned that the poly A site was chosen by the cell, but it's not regulated by external signals.
Which one is not directly chosen by the cell? External signals tell the cell which one to choose. Okay? And how the external signal tell the cell what to do is by the binding of regulatory proteins to regulatory sequences on the mRNA. Okay? So regulation. Okay. All right, RNA splicing. This is the bulk of the variability for us. This is why we only need 21 to 24,000 genes, and we're still a lot more complex than a Daphnia that has, 30 to 35,000 genes, right? It's because for each of the genes that we have, most genes that we have, right, the cell can carry out different types of RNA splicing, and we'll talk about that. Okay? Now, what are the 2 different types of RNA splicing? One is called constitutive RNA splicing. Constitutive is a fancy word that you can use to impress your friends. Constitutive just means always. That's all that it means. In science, we use that a lot. In molecular cell biology, we use that a lot. So constitutive anything means that it occurs all the time, right? It is normal, is the norm. So in this case, constitutive RNA splicing is when normally all introns have to be removed, right? So even without any other regulation, there must be a way to regulate the removal of all introns, at the very least all introns. And so the basic removal of all introns is called constitutive RNA splicing. That only occurs about 8% of the time for us. The majority of RNA splicing is actually alternative RNA splicing, which is the removal of not only introns, so introns have to be removed, but not only introns, but either parts or entire exons are also removed. Again, when parts or entire exon or exons are removed, we refer to that as alternative RNA splicing. I say alternative RNA splicing, I'm missing RNA here because a few years ago we found out that proteins undergo splicing as well. So we can't just say splicing because it's not specific enough. Okay? And so we're already out of date with these slides. Okay? So does that make sense? So in effect, in a small way, alternative RNA splicing has somewhat a similar effect as having multiple polyadenylation sites, right? Because multiple polyadenylation sites allows the cell to make the final mature mRNA of different lengths. So alternative RNA splicing also does the same thing, except alternative RNA splicing can actually remove sequences within the RNA instead of just at the 3' end. Okay? And this happens 92% of the time. And that is the highest number of, highest percentage of alternative RNA splicing that we know of any species on Earth. And you would like to think that this is a major reason why we are so complex, is because of alternative RNA splicing. And I actually have more examples of how powerful alternative RNA splicing is. Yes, sir?
Why wouldn't constitutive splicing happen more than alternative splicing?
That's
It's happening all the time.
That's just evolution. That's how we develop. And like I just said, that alternative RNA splicing is a means for us to have variability and a means for us to be more complex. So why? That should not be a question. It's just how, right? How do we become more complex by having alternative RNA splicing? By having more alternative RNA splicing events than any other species on Earth. But the why is that's evolution. That's how evolution occurs. Seems like I'm not answering your question.
When you said constitutive, I just thought it's like happening all the time. It would be 100%.
No, that's so I explained what constitutive is. Constitutive RNA splicing is the removal of just introns.
Okay.
That's just constitutive.
And alternative is introns or exons.
Alternative is all introns and part or whole exons. So you see, in alternative RNA splicing, you still have to remove all introns. So that's why it's the constitutive part, right? All introns, again, will always have to be removed. Okay. Yes? Okay. Yes, ma'am.
Is there a mechanism for regulation of whether or not there's alternative or
Yes. It's right here. In black and white. It's right here. Okay? So there are all these complexes, and again, there's a picture later on. We won't go into as much detail, but again, conserved structure function, it goes on here too. That there is a complex of proteins called a spliceosome protein that is the equivalent of an RNA polymerase. And in order for RNA polymerase to bind and know which gene to express, what must occur first? Transcription factors must bind to response elements, right? So it's not any different here. Before the spliceosome proteins can form, right, regulatory proteins must bind to regulatory sequences on the mRNA, to recruit these spliceosome proteins. And that's how the cell knows where splicing occurs. Okay, so in terms of constitutive RNA splicing, there are very specific sequences that are recognized. In terms of alternative RNA splicing, there are alternative splicing sites that are found, and we'll talk about all of those. Does that make sense? So again, parallel with transcription. Okay. So let's see. So how? From that question, perfect, it leads on to this. So how does a cell know when to undergo constitutive RNA splicing? From your question, maybe this would help. How does a cell know to undergo just purely constitutive RNA splicing versus alternative RNA splicing? Okay, here's how. When only introns are removed, all introns have what we call a 5' splice site and a 3' splice site. So 5' splice site is found at the upstream of the intron. 3' splice site is found in the downstream of intron, and everything else in the middle is intron. Anything that is outside of these splice sites are coding regions, or exon. So it's very precise. You can't even miss by one nucleotide. Okay? So that's what happened, is that these 5' and 3' splice sites are recognized by regulatory proteins. We know what the regulatory, this actually is well worked out. These regulatory proteins would recognize the 5' splice site and 3' splice site, then will recruit the spliceosome proteins, and that's how, and the spliceosome proteins will cut here, so cleave at both sides, and actually rejoin the exons. This is to ensure that, number one, that the introns are removed in an order, and number two, ensures that the exons themselves are rejoined in the correct order. You will always have exon 1 first, and then exon 2, 3, 4, 5. Even if later on we talk about the removal of, let's say, exon 3, then it's still 1, 2, 4, 5. It's never 1, 5, 3, 6, and they don't jump around, right? So far so good? So that answers that question about constitutive RNA splicing. What about alternative RNA splicing? Yes, sir.
So when you have an RNA that gets constitutively spliced, there's only one polypeptide product, like one possible.
It's always one product. When we talk about alternative, we talk about possibility.
Yeah, no, that's what
But
I'm saying. There's
right,
only one possible
right.
one.
Right, only one possible. So even if there are 10 different ways to undergo alternative RNA splicing, for each nuclear RNA, you can only make one version of that.
Yeah.
Okay, so we agree. Yes. So the answer is yes. Okay. Sometimes I have to talk things out. Again, ESL. So are we okay? Are we okay so far? So in terms of alternative RNA splicing, what happened is that you can either 5' or 3' splice sites can actually found within the exon. And we call these either 5' alternative splice site or 5' cryptic splice site. Probably alternative splice site is easier for you to remember. So you can have either alternative 5' or 3' splice sites within the exons. And let's say you have an alternative 3' splice site within this exon number 2 here, then the regulatory protein can bind to this 5' site, and instead of binding to that 3' splice site, it binds to that. So then not only is intron 1, but half of exon 2 would be removed. So now the final mature mRNA would be shortened by half of exon 2. And that could be, well, we'll talk about what exons represent in a minute. Yes, ma'am?
So if there's alternative splice sites in an exon, is it a given that it's going to be spliced out, or?
No, it's not given.
It's not
So
given.
then it has to do with the regulatory proteins that find those. And the regulatory proteins, in order for them to be expressed or active, then you have external signals. So that's what that picture earlier about which polyadenylation site to choose from external signal. The same with here is that the external signals will tell the cell which splice site to use and which set of alternative splicing, RNA splicing is going to be used. Right? So it's all about regulation. And this is, as you notice, is reminiscent of regulation of transcription, right? So external signal regulates which tissue-specific transcription factors must be expressed and activated, which then regulates which gene needs to be expressed, right? So very much so parallel. So that's alternative RNA splicing. And so the quick example is this, right? You can see that you can have mix and matching of different version of mRNA. Okay? So if I remember correctly, the next one is really powerful. Oh, not yet, but close enough. This is so. What is all this? How do we make it more complex? How about talking about gene expression in different cell types? So a gene, one particular gene, tropomyosin, just one gene. There's only one gene. Talk about this one gene, but it's spliced differently in different cell type. So therefore, tropomyosin, because it's spliced, RNA spliced in differently in different cell types, that means that its function is slightly different in different cell type. That's how we can make it more complex. And again, so how do you regulate it? I'm not going to go through the whole thing, but remember how different cell types are regulated, different pattern of gene expression, different types of regulatory proteins that are expressed, which bind to very specific regulatory sequences, right? So let's look at a something that's not here. Let's look at a T lymphocyte. A T lymphocyte would not have a regulatory sequence that can be bound by regulatory proteins to carry out splicing. So therefore, that's why you wouldn't have myosin even expressed, right? Even if for some reason the mRNA is made, but it won't even have myosin protein expressed because there's no way to splice the introns out. So again, parallel activities here with transcription. Yes, sir.
So it's the exact same gene in different cells.
It's the exact same gene in different cells that are spliced differently.
Okay.
Right. So we're entering yet another level where it's not just the pattern of gene expression, it's the pattern of regulation or splicing. Right. Right. Yes, ma'am.
So in that example that you showed, would that be an inactive gene then?
More than likely is an inactive gene, right. But what I'm saying is that even if for some reason we have a mutation that this tropomyosin gene is for some reason active, right, it wouldn't even get properly spliced because it would be lacking the regulatory region that has to be recognized by regulatory proteins. Right? I think you understand what I'm saying. Okay? Okay, this is the hitting the nail on the head here. There's a protein called DSCAM, which is an adhesion protein, so it allows cells to interact with other cells. There's only one of these genes, and through RNA splicing, one of these genes has over 38,000 possibilities.
Wow.
Yeah, wow is right. This is the extreme. This is like the most extreme that we know of. There aren't any gene that has that many alternative versions. But this one gene allows the cell neurons to have 38 different versions, 38,000 different versions, right, from one gene. The question should be, why? Why would you want 38,000 versions of this one gene, which all it does is that it helps two cells interact? So why do you think that is? Which two cells need to interact? So cell-to-cell specificity in terms of interaction, right? Especially when you talk about neurons. You have however many millions of neurons in your body, in your brain. each neuron only make contact, acting as a signaling cell, right? It receives other signal, and we'll talk about that, but acting as a signaling cell, it will only activate or interact with a small subset of cells, let's say 20 different cells, right? And only a couple of cell types. So then by having a high variation in the DSCAM protein, then your neurons will have a better idea of which cell that it needs to interact with. Okay, perfect example is your family, right? So there are 7 billion people in the world, actually close to 7.5 now, right? Billion people in the world, but you know your family. It's because they have certain features that you recognize. That's the analogy here, right? And the recognition is just looking for features. Everybody has a nose, hopefully. Everybody has eyes. Everybody has a head, but it's slightly different. So that variation that you see in your family members versus everybody else is what the different versions of the DSCAM proteins allow neurons to recognize. That's why you need a lot of variation. Does that make sense? That is a huge concept. I'll give you, later on we'll talk about something, an incredibly huge number as well. I don't know when, I forget when it is, but each neuron, and get this, not all of them, but some neurons can receive up to, this is an incredible amount, talking about external signals, and activating and inhibitory transcription factors fight it out, right? Fighting out of information. Some neurons can receive up to 100,000 signals at any one time. Imagine if you stand in the middle of 100,000 people and they're all shouting at you. Okay, that's an incredible amount of information that is being fed into one cell, and that cell has to determine what to do. Okay, and it's always it's all about upregulation and activation of regulatory proteins that bind to regulatory sequences and so on and so forth. Okay, so it's all about regulation. Yes, ma'am.
They receive the information all at the same time?
At any time, right? So for that example, now that's again an extreme. Most cells still receive several hundreds. Any given cells that you have would receive probably several hundred signals at any given time. So what I just said in terms of the parallel with transcription is you have sequences, or we call it splicing enhancers, and then you have recognition factors that I refer to as regulatory proteins, same difference, okay? And so make that connection, make that parallel connection with transcription. And this is really all that this slide is saying. And then pretty picture of, again, what I already described, right? So 5' splice site is recognized by these splicing factors or regulatory factors, proteins, and 3' splice site is recognized, and then recruitment of the spliceosome. Individual units are called SNRPs. That's what their acronym. Don't worry about what they stand for, but SNRPs, that's how people pronounce it. And then the spliceosome then would splice, remove the intron, and then reconnect the 2 exons. So again, what I just said. Don't worry too much about the drawing or anything like that. I want you to know more of the theoretical stuff.
Kind of like tRNA.
Directly to the okay. So far so good. So another function of the spliceosome, which is connected to, we're still talking about processing, right? But which is connected to, and again, we're only really talking about eukaryotic cells. That the idea of an mRNA will not be transported into the cytosol unless it's completely modified and completely mature. So by being completely modified and mature, it needs a cap at the 5' end. It needs a poly A tail. So therefore, those are used as the two signals to tell the cell that this mRNA needs to be taken out, right? The third is another signal is an inhibitory signal. Okay? As long as the spliceosome complex protein still remain bound to the mRNA, then that tells the machinery that this mRNA is not ready because RNA splicing is not ready, right? So just the reverse from capping and poly A, where the presence of capping and the poly A tail indicates that this mRNA is ready. However, the presence of the spliceosome complex indicates that this mRNA is not ready to be transported. Just the opposite. And certainly, even if you don't have the spliceosome complex, but for some reason the cell forgot, and it can happen, to remove a certain intron, the presence of an intron, whether or not a spliceosome complex is bound, is also unacceptable. Okay? It's also an inhibitory signal preventing the cell from delivering the mRNA into the cytosol. So there are multiple signals, right? Both pro and anti. Right? Okay. So this is another important kind of general ideas about exons and RNA, alternative RNA splicing, and so on and so forth. In general, exons, let's say a small RNA, a small mature mRNA would contain probably about 2 or 3 exons. There are some, though, that would contain hundreds of exons, right? So the size of the mRNA varies, the mature mRNA varies, and of different mRNAs, not just from the same gene, but different mRNAs vary, and the size of proteins vary, okay? However, so then come back to the question of, well, why is exon 1 exon 1? Why is exon 2, and so on and so forth, right? Each exon roughly is going to be translated as a unit, right? As a block of amino acid sequence that, when folded, would form what we call a domain. So each exon or each group of exon, let's say you have a protein that contains 10 exons, right? And it has 2 domains, a binding domain and a catalytic domain. Then the binding domain would be dictated by exons 2, 3, and 4. And then the catalytic domain is embedded into exon number 8 and 9. Okay? So either individual exons or groups of exons would be expressed into an amino acid sequence and fold into what we call a domain. And a domain is just a region within the protein. Analogy would be your home, right? You have a house, which is analogous to a protein, but you have different rooms. And the different rooms have different functions, right? So each room is analogous to a domain. Okay? And some rooms are bigger than others. Therefore, some domains are bigger than others. Could be bigger because with actual exons, each exon is bigger, or bigger because you have more than one exon that contribute to the formation of that domain. So when you have alternative RNA splicing, if you build a house, right, you can have a house with 2 rooms and 2 bedrooms, 2 bathrooms, versus a house with 3 bedrooms and 2 bathrooms from the same construction company, and all they did was they just removed 1 room. So that is an analogy to alternative RNA splicing, where you have the same blueprint, you just, to make a different version of it, you just remove something, right? And by removing an exon or removing a part of an exon, then you are removing either the whole domain or a part of a domain. By doing that, what are you doing? You're changing the function you're slightly changing the function of the protein, but still, the protein still remain as is. What might be an example of alternative RNA splicing, and the proteins still have the function, but different? How about antibodies? Perfect example. Antibodies. Antibodies can be in two different forms. So actually, no, antibody, that's more of an example of different polyadenylation sites. Why don't I don't give you an example? Because the example I think of is pretty complex, but you kind of get alternative RNA splicing. Alternative RNA splicing then allows the cell to remove either a part or whole domains. Therefore, the function of a protein changes slightly. Exon shuffling. I don't think I'm going to talk about exon shuffling. We're good. We're good. Okay, so that's the idea of domains. So what I just said, domains, right? Domain 1, 2, 3, forming that might be the binding domain, that might be the catalytic domain, right? And let's say alternative RNA splicing and you remove domain 3, then you don't have a binding domain, you just have a catalytic domain. So what does that mean? That means that maybe this enzyme normally would be specific for a certain substrate, now it's general. That is kind of sort of a rough example of what alternative RNA splicing might be able to do for a protein. In terms of domains now we're kind of diverging slightly. So not talking about alternative RNA splicing, but talking about domains, right? When we talk about domains, we go back to talk about conserved structure and function and the correlation between structure and function. If a domain, if a function is important, then it's conserved. And the structure to gain that function should also be conserved, right? That's the idea. And the structure then equals domain. So for instance, elastase and chymotrypsins are both what we call proteases. Proteases are enzymes that cleave proteins, that cut proteins. Okay? Both of them cut proteins, so the catalytic site, the site that actually does the cleavage, needs to be conserved because that's an important function. Therefore, because it's an important function, the structure is conserved. As you can see, it's drawn here. It looks about the same. And if the structure is conserved, therefore, the domains are conserved, right? So the idea is that these two proteins actually came from the same origin. So through gene duplication, independent DNA mutation, right? Then you would have two different enzymes still having a major function, which is cleavage. What is different? Difference is, let's say, which protein that it cleaves, where it cleaves. This might be different. But the main function of cleavage is the same. And because that main function is the same, the structure has to be the same, right? It has to be conserved. So even through gene duplication and independent mutations, the domain, the exon that yield this catalytic domain would not have been mutated, right? Does that make sense? We're combining all kinds of stuff. I'm doing it for you, so that's good. But you have to understand what I'm saying, though, right? Even though I'm doing the thinking for you.
So, the exon that leads the function doesn't change ever.
Right. You would think that it doesn't change, right? Because if it does change, then the function would change.
So it couldn't work anymore.
Or it works better, or it has a slightly different function. Instead of cleaving proteins, it might cleave lipids. That's an extreme example, or probably never happens, but that, if you change that exon radically, then you would have a base different
Change
function.
of the function.
Uh,
But if you want to keep the function, then you have to keep the.
It has to be conserved, right?
Has to be
Right.
conserved.
So function structure, right? Conserved function means it's an important is important, but in order to conserve the function, you need to conserve the structure. In order to conserve the structure, you better leave the exons alone. So last thing, and then, right? You know what? This leads to a whole set of new things. So we're done. Have a good today's Tuesday. So have a good day, and I'll see you